रखने का अधिकार नहीं। क्लास होगी स्टार्ट। डम्मन ले नहीं बनाई मैगी। पानी दो मैनू तो बस।
हो जा सीधे बैठ टेबल ना मार।
पानी साफ नहीं हैगा. Very good evening, everyone. Very good evening. Very few students are joining us at the moment. So just wait for 2 or 3 minutes, then we will start. I have to do a lot of things today. So we're going to start with our topics straightforwardly. Hello, 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 everyone. Thank you for subscribing the channel also. It really helped me actually. I have seen some straightforwardly um, quite jump. Oopsie doopsie. Sorry, sorry, there was a misconnection. I hope it's all back now. Good, good. So we can start now. We'll not waste much time. So we will start with very basics about molecular biology, then we go to advanced level. So today the task is to know about cell structure, about DNA, what DNA is made. Then we will slowly start with RNA structure, and then transcription, translation, yeah, codo- your what is that? Transcription, translation. Even I forgot the name. We will know. We will know. So yeah, the central dogma. I just misplaced that name. Central dogma of the biology. So we'll listen. We will discuss mainly about the key main players of molecular biology. That is your DNA, RNA, and proteins. Okay. Coffee बनाते हैं मैंने. Coffee, coffee, coffee. Not okay. I said coffee. So students, let's start with a fresh mind, with the fresh sessions of today. So this is an analogy discussed here. Basically, cell is like a factory, right? So how can cell be like factory? So as we know, in the factory, we have many different tasks being completed in the same building. like support, shelters, workers, build goods, maintenance, crew cleanup messes, ship departments, goods ready to leave factory, and so on. So a cell can be thought as a factory with different partners, each performing specialized tasks. So here we have Golgi vesicles, nucleus, centrioles, microtubules, cytoplasm, cell membrane. So some like nucleus is a CEO, cell membranes are like security guard, right? Mitochondria is a powerhouse of the cell. So each part has its own component, and they are working like a factory. So like a cell wall, which is a security gate that protects the factory and helps keeps it safe, so that there is no problem in going any bad thing towards your cells. And same way, cell walls does not allow anything to go into the cells, so cell wall do the same thing. And then cell membrane, this is the front door of the factory, which regulates who will enter, leave the building. So cell membrane does the same thing for plants and animal cells, so like this. In the nucleus, this is a big boss, CEO. The boss controls all of the activity inside the cells. The nucleus does the same thing for plant and animal cells. So this is the boss under which the nucleus is working as a main part of it. And cytoplasm, which is a factory floor, on the floor of the factory, which has all the workers, machines, and equipment on it. So like cytoplasm, which holds all the organelles in the plants and animals. So they are maintaining the workers, machines, all the places. The movement of your like, let's say after DNA is being transcribed into the mRNA, then mRNA is going into the cytoplasm itself, where it's changing into the protein. So in the cytoplasm, your mitochondria, Golgi body, all these organelles are present. And ER, which is like an assembly line where the workers do their work. So items move along the conveyor belt to different parts of the plant. So ER moves items to different parts of the cells in the same way. So this is like conveyor belt here, endoplasmic reticulum. So like if a Coca-Cola factory or any boot machine is there, or anything which is being manufactured at a large scale, so they all are going via conveyor belts. So ER is acting like a conveyor belt here. Then ribosomes, so workers on the assembly line, they're responsible for building the goods in a factory. So ribosome assembles your proteins. Here you can see. Then vacuoles, they are like break rooms. In a break room, you have a storage for food items and places to get rid of trash there. So everything is taken care within that part. So they hold the waste. So same way, vacuoles are the ones which hold the waste and stores waters in plant and animal cells. So which cell has one large vacuole? So like this. So lysosomes are like maintainers, janitor crews. So in a factory, these guys break down, clean up and repair items in and around the factory. So lysosomes do the same things in the cell. So they care of any if things got broke up or if anything needs to be changed out. So these lysosomes do take care of that. So mitochondria and chloroplasts, power stations, they supply energy to the factory. And mitochondria and chloroplasts, they do this for the cell. So this is the example of the factory. So this is your ribosome here. This is your mitochondria. This is endoplasmic reticulum. This is your nucleus, Golgi apparatus, restricted area, membrane. So this is a factory where your cells, things gonna come in and out. Then ER is attached to your nucleus where chromosomes are being produced. Then mitochondria as an energy producer here, the fuel generator, working on the conveyor belt, these ribosomes, and producing proteins. Then this chromosome is attached to now your nucleus to Golgi apparatus also for the waste product producing. So in this way we can see very nicely the Golgi and proteins, ribosomes, mitochondria, ER, nucleus, chromosomes, cell membrane, all are working in a factory in a very nice analogy. This is not required for you. So that's it. So just want to give you a brief touch about there's the link, Urvi. It doesn't came with the picture today. Yeah. So that's the very basics actually about the cells. And what about the cell size actually? In a brief, I want to take you to this animation now. So here we can see a size of coffee bean with a 12 by 8 millimeter. This times regulator point with 12 point grain rise of about 8 by 2.5 millimeter. So what we're gonna see is that now that being size of this, and we will go to the carbon atom, which is a picometer size at 10 raised to power minus 12. I hope you know nanometer, picometer, micrometer. So we will now zoom in. So this is grain of salt, amoeba, which is 500 micrometer, then paramecium 210 by 60 micrometer, human egg 130 micrometer, photoreceptor 2.5 to 100 micrometer. You see human egg is 130 micrometer, where sperm size is about 60 by 5 micrometer. So skin cell 30, red blood cell 8, chromosome X chromosome 7, bakery is 3 by 4, then mitochondria 4 by 0.8 micrometer, E. coli 3 by 0.6, lysosome 1, then measles. This was all about micrometer. Now we come to viruses. Viruses are in the size of nanometers. Even a COVID virus is a nanometer size. 220 nanometer, 130 nanometer, HIV virus 130, coated vesicle 90 nanometer, phage 70 by 200, hepatitis virus 45 nanometer, rhinovirus 30 nanometer, ठीक, ribosome 30 nanometer, antibody 12 nanometer, transfer RNA 7 nanometer, hemoglobin 6.5 nanometer. Now we go even more deeper, phospholipids 0.9 to 3.4 nanometer. Now comes the picometer. Now after nanometer, now comes picometer. These are nucleotides like adenine, 1300 by 760 pm picometer, glucose 900, water molecule 275, yeah, methionine 1100 by 700 picometer, which is an amino acid, and at the end your carbon is around 340 picometer, which is the basics of your making the whole thing. So from your picometer of carbon atom with the adenine, glucose, phospholipids, antibody, all the viruses, nanometers, then all the eukaryotes in the micrometers, yeah, skin cells, body cells in micrometer, then grain salt, sesame seed, grain of salts in millimeter. So we can see from this perspective how things could be also in so so deep, so could be so tiny, and what we are seeing as a cell, right? So the cell will have so many things also. So all in all, the main reason to show you this comparison is initially we talk about today cell as an analogy of the factory, and then corresponding to that cell size and scale, how everything within the cells is been set up very nicely, and how eukaryotes, prokaryotes, they are coming together. Okay, so this was about it, cell size. So I will start, I will not because I tend to forget to share things later, so I will keep sharing things whatever we will be discussing here at once, so all these things will be shared same time. So you will have by the end, there is a thing called here, if you open your this thing, these files, so you will have lots of files with each day, with each month, being divided, and then you can find them. Yeah, some links are there: files, media, members, and so on. Okay, so two things are over. Now next is structure of DNA. So you all might be aware of, you are in masters, PhD, some are MBBS, some are in class 12. You all been through this part of structure of DNA. But before we start about anything about molecular biology, our basics need to be clear. Our basic understanding of molecular biology needs to be very much clear. Yeah. So in this part, you will get to know about the concept of DNA, its structures, packaging into the nucleus of the cells. You're able to demonstrate the concept of central dogma in molecular biology. So first thing first, history. So nucleic acid was first isolated by Friedrich Miescher in 1869. Then Friedrich Griffith, he discovered the factor in disease bacteria, which can transform harmless bacteria into deadly bacteria. Then Rosalind Franklin got very fine with X-ray photograph of DNA. Then Watson and Crick described DNA molecules from Franklin X-ray. It is 1953. Then Chargaff rules, that is purines are equivalent to the pyrimidines. So mainly your Watson and Crick are the one who have discovered this DNA molecule, and they also got Nobel Prize winner for the same. So what DNA structure consists of? It's a double helix structure with two chains having sugar phosphate background on the outside and nitrogenous base inside. So molecule of DNA is made up of millions of tiny subunits called nucleotides. So like this. And prokaryotes includes your nucleoid and plasmids, and eukaryotes includes chromatin of nucleus. So DNA has three main components, that is deoxyribose, a pentose sugar, base, there are four different ones, and phosphate. This is the phosphate, deoxyribose, and base. So these bases could be four type A, T, C, G. Mainly DNA, it's all made up of A, T, C, G. The difference in this A, T, C, G, you, me, our parents, our brothers, our sisters, in future our kids, we are all different. Dogs are different, whales are different from human being. All organisms, species, they're all just different because of A, T, C, G. That's the main idea behind the differences in this world. A sparrow, a peacock, a vulture, a fish, a whale, a chimpanzee, a human being. Within human being, let's say a person with the dark race, a person with the white race, a person with the brown race, a person with the Asian race. So all these people, all these things that I have said, they are all just differences of ATCG, nothing else. Yeah, we have made these differences, we have given them the name, we have created the differences in our eyes, but at the end, we are all made up of DNA. That's it, and the main difference is ATCG. So here the RNA and DNA, the main difference is of the ribose sugar. The ribose sugar is made up of these 5 carbon atoms. So in the second carbon, you have a hydroxylic group, right? In this hydroxylic group, your OH group is there. But in the DNA, there's a deoxyribose sugar, the second carbon, the OH group is removed. That's why it's called deoxyribose nucleic acid, and the RNAs are called as ribose nucleic acid. So they are divided into two groups, these bases, pyrimidines and purines. So pyrimidines, they are made up of 6-member rings, thymine and cytosine. So furthermore, purine is made up of 6-member ring fused to a 5-member ring, which adenine and guanine. So here we have adenine, and here we have guanine, and here we have thymine and cysteine with only one 6-member ring, right? Further, these long chain of your DNA, it's made up of you have a phosphate group, sugar, phosphate, sugar, and towards these sugar group, you have different purines and pyrimidines attached. So like adenine, two-membered ring, guanine, two-membered ring, cytosine, thymine, and so on. So here we have nucleosides and nucleotides. So nucleosides are mainly the one which are without the phosphoric acids. So they have base and deoxyribose sugar. So if you have a nitrogenous base adenine, and towards this adenine group you have attached this deoxyribonucleoside, that is deoxydeanosine. Then guanine, deoxyguanosine, cytosine, deoxycytidine, thymine, deoxythymidine. Right? So deoxy, then if you add phosphoric acid to deoxyribonucleoside, it becomes deoxyribonucleotide. So you have deoxyadenosine monophosphate, deoxyguanosine monophosphate, deoxycytidine monophosphate, deoxythymidine monophosphate. The abbreviation would be DMP, DGMP, DCMP, and DTMPs. So nucleotide structures, they are formed by the condensation of pentose sugar, phosphate, and one of the four bases, that is ATCG. So here phosphate, sugar, and nitrogenous base pair. So and these two sugar and phosphate group, they are linked by the phosphodiester linkage. They both are attached by the phosphodiester linkage. So DNA double helix and hydrogen bonding, they are made up of two strands of nucleotides that are joined together by hydrogen bonding. So hydrogen bonding occur as a result of complementary base pairing. So adenine and thymine pair ups, cytosine and guanines, they pairs up A with the T, C with the G. A pairs with T with a double bond, and C pairs with the G as a triple bond. So each pair is connected to the hydrogen bonding here. So hydrogen bonding occurs between one pyrimidine and one purine always. So this is very nice example of antiparallel strands. So here on this thing, so here as we know, we count this sugar structure from here: carbon 1, carbon 2, carbon 3, 4, and 5. So the fifth carbon, your sugar is ending with the phosphate group. So that's why it's a 5' end. And here, and the third prime carbon atom, 1, 2, 3, your sugar is ending. So the 3' end. Another side is 5' end, another opposite side 3' end. So that's why it's an anti-parallel structure. So both are going in anti-parallel way. And in between to your sugar group, you have a nitrogenous group attached like adenine and thymine. And then like this is, and then your guanine and cytosine here. So the strands run opposites of each other, and this adenine and thymine, they are making double bond, and your guanine and cytosine, they are making the triple bond here, right? So adjacent deoxyribonucleotides are joined in a chain by phosphodiester bond, which links 5 carbon deoxyribose sugar of mononucleotide unit with 3 carbon of deoxyribose of next mononucleotide. So this is 1', 2', 3', and 4', and 5' end. And yeah, so between these two, this phosphate here is making a ester bond. We call it phosphodiester bond because it's making two ester bonds actually. One ester bond here, one ester bond here. So that's why it's called phosphodiester bond. So two chains wrapped helically around each other with sugar phosphate outside and base inside, and hydrogen bonds between the base pairs. So two chains are complementary to each other and anti-parallel strands with them. The coiling of DNA is right-handed, and one complete turn occurs every 34 angstrom. And 10 mononucleotide occurs per turn. Helix has two groups, that is major and minor. So what is the meaning of right-handed? It means this we have discussed all things above 4 points. So right hand means the moving of your DNA is in right-handed. like if you have a right hand, if you move your all four fingers towards yourself, they will move in the right direction, right? So that is means right-handed direction. One complete turn means it includes your major and minor groove. If it has these two grooves together, the size is 34 angstrom. And within this this is called as one complete turn. And within this one turn, there are 10 mononucleotides are present. And this type of DNA is the most common DNA, and it's called as bDNA. And there are three types of DNA: aDNA, bDNA, and zDNA. We will discuss them in upcoming slides. So here we can see the length of one turn having the major groove and minor groove of size 3.4 nanometer, that is 34 angstrom. Yeah, and minor groove, the size is around 1.2 nanometer, major groove 2.2 nanometer. The major groove description is that the turn is going on, it's more deep, yeah, more inside, and the minor groove is more narrow here. Yeah? And the diameter of DNA is 2 nanometer. So it's moving from 3' end to 5' end like that. And two parallels are anti-stand with each other with A, T, C, G. So this is a ribbon model, and this is a space filling model. So features of DNA double helix. So two polynucleotide chains, they coil together to form right-handed helix. So two chains are anti-parallel. So both runs from 5' end to 3', but their orientations are opposite. So nitrogen bases of opposite chains pair to one another and form H bonds. And the base pair stacks on each other located inside the helix. And each complete turns is of 3.4 nanometer long and contains 10 bases. And each turn along the molecule contains one major and one minor groove. Right? So there are three types of DNA, that is aDNA, bDNA, and zDNA. So aDNA is right-handed double helix, compact with 11 base pairs per turn, that is 2.3 nanometer in diameter. bDNA is found in mostly all living systems, right-handed double helix, compact with 10 base pairs per turn and 2 nanometer in diameter. And left-handed double helix called zDNA because of its zigzag structure and compact with 12 base pair per turn and 1.8 nanometer in diameter. Yeah, so you have understood the basic difference between your A and B. A is more like it has 11 base pairs, B has 10 base pairs, and zDNA has 12 base pairs in one turn. It has a zigzag structure. Here the diameter is a bit thin, that is 1.8 nanometer, B is 2, and A is more wider, it's more thicker, that is 2.3 nanometer. Yeah, students? Yeah, you will get all the PPTs, everything you will get it, don't worry. So review article, Ramesh, Ganipasiti, it has to be, you can be also write single, but I will prefer in work in groups. At least maximum is 4, minimum, yeah, 1 single. So students, that was, we will continue now. I just came back to see if everything is fine, streaming is fine, lectures are going fine, and we continue back. So this is the, now before we talk about any further things, so I want to talk about this bDNA. Here we can see this is the major groove, this is the minor groove, these are the nucleotide base pairs. So in aDNA it's opposite, the minor groove is more deeper and the major groove is more like this, right? And there is a protein induced deformability parameters. So sometimes there is a nitrogen, your nucleotide base pairs, they have an angle from each other, they make an angle, and that angle is called as tilt. And the distance between two nucleotide base pairs, it's called as rise. Distance between two nucleotide base pairs is known as rise, and tilt is the angle between your two nucleotide base pairs. So these things are never discussed in most of the things. So here also A and B DNA, it's a right-handed helix, and the Z DNA is left-handed helix, and here we can see it's more thinner. This is an average size, the in between, and this is more broader, A DNA. So that's the major difference between your A, B and Z DNA. And here this is also the Z DNA zigzag shape. This is normal, and this is very gooey. So this is the major difference. So let's discuss a little bit about A, B and Z DNA. So overall proportion, this is a short and broad, this is longer and thinner, this is elongated and slim. Why they are named like that? Because the rise per base pair, that is the distance between two nucleotides, 2.3 angstrom, 3.32 angstrom and 3.8 angstrom. And this is helix packing diameter, 25.5 angstrom, 23.7 angstrom and 18.4 angstrom. And the helix rotation sense, right-handed, left-handed. Yeah, base pairs per helix repeat 1, 1, 2, and this is the further helix axis location major throughout the base pairs minor groove, and the tilt form is plus 19, the angle between the two nucleotide base pairs, plus 19, minus 1 to minus 9. And helix axis location and major groove proportions, extremely narrow but very deep, wide with intermediate depth, flattened out on helix surface, and the minor groove is very broad but shallow, narrow with intermediate depth, and extremely narrow but very deep. So that's the major forms of DNA. So we have discussed so far the main structure of your DNA, purines and pyrimidines, how this DNA is made up of, various types of DNA. We have discussed the cell structure. We have discussed how the cell size matters, all the components, the size that matters. And the second thing, now next we're going to discuss a little bit about further classification. It's very simple, linear and circular. Linear is found in eukaryotes and circular in prokaryotes. This is in the linear in which both ends are free. With the circular it will not have a free end. So like that, right? So this is very small, very nice 10 question answer. So what we have discussed so far. So DNA stands for deoxyribonucleic acid. Shape of DNA is helix, double helix structure. Who established? Watson and Crick. Adenine pairs with guanine. The sides of DNA ladder are deoxyribose and phosphate. Guanine always pair with thymine. Complementary sequence will be T, T, A, C, G, T. Two sides of DNA are held together by hydrogen bonds. DNA is composed of repeating subunits called nucleotide base pairs, I guess. So what are the four bases that makes your adenine, thymine, guanine and cytosine? So that's very basics. I hope you have gained an overview of structure and function of DNA as a genetic material. You can correlate now downstream topics also in further upcoming replication, transcription, translation. So we will have these lectures now from now onwards. So some references here, and that's it. So structure of DNA. All good students so far? Yeah, I wanted these questions to be answers by you actually, but I want to make sure that we can't. Oh, the battery is very less. I wanted to make sure that we continue with the pace. So I have shared the questions also. You can go through them, and then we can think about it. Yeah. So no worries, no worries. So next part I want to discuss now is RNA. So we have discussed DNA very nicely. We have discussed the structure of DNA, types of DNA. And now comes the RNA part. Good. I need to remove my pictures. I was told that it's good to put pictures of yourself, but I feel really awkward to have my pictures. Anyhow, so yeah, molecular biology, it's basically study of molecular underpinning of process of replications, transcription and translation of the genetic material. So it has animal cells, plants, microorganisms, molecules, populations. And within these different 6 domains, there's a different studies are named as, like genomics, enzymes, extremophiles, microbial pathogenicity, plant development and defense, imprinting, cell cycles, molecular neuroscience, animal development, molecular entomology, mathematical biology, and so on. So basically, molecular biology is a study of molecular underpinning of the process of replication, transcription, and translation of the genetic material. So this field overlaps. So much of the work in the molecular biology is quantitative and has been done at the interface of the molecular biology. So in 15s and 60s, molecular biologists have learned to characterize and isolate, manipulate the molecular components of cells and organisms, include DNA and repository of genetic informations. So RNA, a close relative of DNA, proteins, and major structural of enzymatic type of molecules in the cells. So this is the components that are involved in molecular biology, that is DNA, RNA, and your protein. Yeah. So I will not discuss about DNA here anymore. So we have discussed the DNA very briefly so far. So you can have it look there. It's all things that we have discussed. Now comes the RNA actually. So RNA is a biologically important type of molecule that consists of long chain of nucleotides units. So each nucleotide consists of your nitrogenous base pairs, a ribose sugar, and phosphate group. So instead of deoxyribose sugar, we have a ribose sugar. So here we can see it's a single base pair, right? And then it's become a double helix for some 3 base pairs and having a hydrogen bond between them. And then it's again become single base pairs. Then it's become, yeah, at the corner you have a sugar phosphate backbone, and in this one you have your bases. Then the double-stranded RNA. So double-stranded RNA is RNA with 2 complementary strands and similar to the DNA found in all cells. So double-stranded RNA forms the genetic material of some viruses, that is double-stranded RNA viruses. So there are types of RNA, messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA, they codes for proteins, they have all organisms, and ribosomal RNA, their translation for all organisms, and transfer RNA also for the translation and present in all organisms, tRNA. In the post-transcriptional modifications, we have small nuclear RNA, snRNA, yRNA, and telomerase RNA. In the regulatory RNAs, there are antisense RNAs. Then according to different types of RNA, let's discuss the first type. This is messenger RNA. So messenger RNA, which carries information about a protein sequence to the ribosomes, and they are the protein synthesis factory in the cell. So they are coded for every 3 nucleotides correspond to 1 amino acids, and eukaryotic cells, one pre-mRNA has been transcribed from DNA and it is processed to mature mRNA, and this removes its intron, non-coding sections of the pre-mRNA. So you have your bases here. Yeah, you have bases, you have your DNA, right? And this is your coding strand, CAATGGCG, and your messenger RNA is being prepared here. This is a coding strand, this is non-coding strand. So in front of C is G, A is U, because here instead of thymine, U will produce in messenger RNA. So instead of T, U will produce here, that is uracil. So messenger RNA is then exported from nucleus to the cytoplasm, where it is bound to ribosomes and translated into corresponding protein from the help of tRNA. So this line that I said, the mRNA then exported from the nucleus to the cytoplasm, where it is bound to ribosomes and translated into its corresponding protein formed with the help of tRNA. So in this, in prokaryotic cells which do not have nucleus and cytoplasm compartments, mRNA can bind to ribosomes while it is being transcribed from DNA. Yeah. So this is the main thing that we will discuss now with the help of animation. That how transcription, how translation, all these things works and plays together. Yeah. So let's see what is DNA and
The instructions for building all living things, whether a fish, a bacterium, a tree, an insect, or a person, are found in a molecule called DNA. DNA is made up of 4 tiny building blocks. Each building block has a backbone and a chemical base that is represented by the letter A, C, G, or T. DNA molecules have 2 strands that are joined together through complementary base pairing. A pairs with T, and C pairs with G. These building blocks function together in larger units called genes. Each gene is a set of instructions for building a specific protein. A complete set of genes is called a genome. A genome is a set of instructions for building an entire organism. Every person's genome has the same genes arranged in the same order, but small differences in the sequence of the bases in our genes make each person unique.
Yeah, so that's what I said initially, that why we are all different, why everything is different. Now this is very interesting, same gene, different organism, that you will have a different organism, but you have to put the same gene, you get the same function out of it. Yeah, so for example, there's spider webs. Excuse me. So into the spider webs, spider, you put silk gene, they will produce lot of silk out of it. Same way, the glowing gene GFP you produce into the fish, they will be start glowing in dark. So let's check this.
These may look like ordinary fish. What you can't see is that they have a gene in them that codes for green fluorescent protein. But shine a blue light on it, and GFP emits a bright green glow. What might surprise you is that the gene for GFP came from a jellyfish. GFP was first discovered in jellyfish over 50 years ago. Since then, scientists have been using it to study all kinds of biological processes. What's amazing is that this gene from jellyfish can be put into just about any living thing: animals, fungi, plants, bacteria. They can read the DNA code of a gene and build GFP that is identical to the jellyfish protein. That same concept applies to tons of other genes too. The gene that spiders use to make silk webs, for example, can be put into yeast cells. Harvest the silk protein that the yeast cells make, and you've got the raw material to make a strong, lightweight fiber. In fact, a company is using this approach to bring yeast-grown spider silk fabrics to market. How is it then that fish and yeast can read DNA from a completely different organism? It's because, despite their differences, there's something fundamentally the same about how all living things read genes to build proteins.
Yeah, very interesting, very good point. That's actually all molecular biology is all about. So next point. Okay, this is my dog, Candy. A little bit about proteins. No, Candy later.
Proteins carry out most of the body's functions, and they make up the bulk of its structure, from hair and fingernails to bone and muscle. In fact, nearly everything in our bodies is made from or by proteins. The instructions for building proteins are found in segments of DNA called genes. Proteins are made of long chains of building blocks called amino acids. There are 20 amino acids in all. Put together in different combinations, amino acids form proteins with a broad variety of shapes. This diversity of form gives proteins their many functions. The proteins a cell makes determine what it can do. Our cells are constantly at work, producing the thousands of different proteins that keep our bodies functioning.
Yeah, that's how proteins are working out. Now I'm gonna play the last animation of today.
Transcription and translation are the processes that all living things use to read the information in DNA and build proteins. Each gene in a cell's DNA codes for a specific protein. During transcription, cells make a copy of a gene out of mRNA. During translation, they use that mRNA copy as a blueprint to build proteins out of amino acids. These proteins perform the tasks that make living things function. The flow of genetic information from DNA to mRNA to protein is central to life.
Oh, it's got stopped somehow.
Transcription and translation are
So let's take one gene here.
inside the nucleus. RNA polymerase attaches to a gene. It separates the two complementary DNA strands, exposing the gene's unique sequence of bases.
So the A pairs with U, G pairs with C, A pairs with U, T pairs with A, C pairs with G pairs with C, with A, U, G, A, and so on.
When the end of the gene is reached, transcription is complete. The new strand of mRNA passes out of the nucleus and into the cytoplasm, where translation takes place. The instructions in an mRNA strand are read in groups of 3 bases called codons. Each codon must be translated, one after another, in order to build a functioning new protein. The key structures in that process are the ribosome and transfer RNA, tRNA. Each tRNA molecule has its own anticodon and a matching amino acid, the building blocks of proteins. To begin translation, a tRNA molecule coded for the amino acid methionine docks with a ribosome. The ribosome attaches to the end of the mRNA strand. It slides along until it finds a start codon, AUG. Through complementary base pairing, the start codon pairs with the anticodon on the tRNA molecule, signaling the beginning of the protein instructions. The methionine molecule is now the first amino acid in the new protein. The ribosome shifts to read the next codon. Each codon is recognized by one of 20 different tRNA molecules. Each type of tRNA carries a different amino acid. The ribosome continues moving along the mRNA strand, one codon at a time, matching it to a complementary tRNA. It adds each amino acid to the growing protein in just the right order. The sequence of amino acids determines the final shape of the protein and the function that it will carry out. Give translation a try.
So let's see. ACA. So which tRNA? Is it UGU threonine? Yes. GCA. CGU alanine. Yes. And this way it's keep going on.
When the ribosome reaches a stop codon, it detaches from the mRNA strand. The chain of amino acids folds into its final form and the new protein is complete. This protein, aminoacyl tRNA synthetase, will now attach the right amino acid to the proper tRNA molecule, charging the tRNA so that it can participate in protein synthesis. Each type of amino acid has its own enzyme. The incredible processes of transcription and translation enable cells to carry out all of the functions necessary for life.
Yeah. That was all about translation, transcription. That is your central dogma of life. That was the key point today. That is how the central dogma of life is playing big role in the field of molecular biology. So I hope I will stop it here. We are done with the mRNA till now, I guess. In the next, I will discuss tRNA tomorrow. We will start from transfer RNA, right? And students, I just want to make one request to all of you. I want that classes to be taken in the morning also, because I might be leaving for some holidays for 1 month or so. So it's my humble request to all of you, if we can take one classes 1 hour in the morning and 1 hour in the evening, or if you want, you can take class with me from 4 to 5, 5 to 6, but that will be too much for 2 hours for you to take all molecular biology at once. These lectures will be recorded. You can come to them every time. If you can't attend them, that's not a problem. But please discuss in the group that if you can make one session in the morning, like let's say 8 a.m. in the morning, 7 a.m. in the morning, 8 to 9, or and then we have one session 5 also, so that we can make these classes of in 20 days, we can finish with them with the 10 days. It's my humble request, though they promised you to make it to 20 days, but I have some family emergency. I need to leave from home actually. You have a colleges, Momita Jaha. Okay, fine, agreed. But how about 7 in the morning? It's 7 to 8. You have your school, yeah. Okay. So it seems very difficult for you in the morning. Okay, no problem. We will keep our classes like this at 5 p.m. so far. And we will have our classes over the weekend also then, Saturdays and Sundays. So that at least, no worries Vaishnavi, no worries. We keep our classes at the moment 5. So tomorrow we will discuss about tRNA and ribosomal RNA, and then we will take the story to the translation and transcription, and then little bit about replication also, how replication is done. Then we will take little bit about the tools in my molecular biology. So tomorrow it's gonna be more interesting. We will discuss from PCR, the PCR virtual lab, that how PCR is done in the lab, right? And if we have time, we can go for DNA isolation. So that's it, students. You might have class now of the vaccine, right? So I will share this link 5 minutes before or 10 minutes before. I will try to share it 10 minutes before, so that at least 50 or 60 students could be joined with me, and then we can have this very nice successful classes like this. Yeah? If you have any questions, I've shared 3 videos um, over this Telegram group today in the morning. So something about Ikagai, about how to write a resume, how to go for abroad, or if you have any other questions, write me in private message or write me in the group. I will be happy to answer them. I will try to make one video so that every could be benefited out of it, and everything will be done. Yeah, blotting, Soumya Mukherjee. Blotting will be done in detailed manner. We will do southern blotting, northern blotting, western blotting, um, agarose blotting, electrophoresis, gel electrophoresis, agarose electrophoresis. We will do 2D gel electrophoresis. I don't know how many, paper electrophoresis, zone electrophoresis, capillary electrophoresis, horizontal electrophoresis, vertical. There are so many. So don't worry, your blotting part will be covered. We will do that in detail because that's the most essential part in molecular biology. Believe me, if you want to be a molecular biologist in your future, you tend to do a lot of blots, and these blots tend to run for 2 days. So you have to be very smart while running them. We will discuss. I will share some tips also. Yeah. Okay. Thank you. See you tomorrow. Yeah. Until then, wish you all nice evening. Good luck and nice Wednesday. Take care. Bye bye.